Walaikum salam. Yeah, good. Good morning to all of you, and we'll continue with our this nosocomial infections that we left last time. But this nosocomial infection will be much more concentrated on Staphylococcus aureus. So all of you know about this nosocomial infection. We are continuing since a few classes. So uh, dear student, be log in before 11 a.m. of the class because it's very disturbing when I am on the teaching mode and you are coming and joining. It's getting disturbing to me and other students as well. So you should come online before 11 a.m. so that you could join the full class. So there are four factors that can influence the nosocomial infections, and these are the microbial factor. This is the main factor. The patient susceptibility, this depend upon the immunity of the patient, how much the patient is susceptible to the nosocomial infection. Environmental factor, that is temperature, humidity, all these things, and bacterial resistance. So whether the bacteria is resistant to chemicals, resistant to antibiotics, and resistant to other factor. If it is resistant, it will spread the nosocomial infection much more rate than anything else. So the story of Staphylococcus aureus. Staphylococcus aureus was first described by British surgeon Sir Alexander Ogston in 1882. So it's been uh, more than 240 years since the this discovery of Staphylococcus aureus by British surgeon, and he described this bacterial strain and its role in sepsis and abscesses. So, how Staphylococcus aureus do sepsis and abscesses uh, when it infect any patient or any person, and it is one of the most dangerous community acquired and hospital acquired pathogen. So Staphylococcus is one of the most dangerous community acquired and hospital acquired pathogen. So how sepsis happens? So this is Staphylococcus aureus infection sites, and this is abscesses. Everybody might have idea about this kind of infection, right? So in their lifetime, sometimes everybody get across this in body or in face. Sometimes like pimple, all these things. So what is sepsis? Sepsis is excessive bacteremia. That means bacteria growth in blood which causes blood poisoning. If the blood is poisoned by Staphylococcus aureus, it is very dangerous. So in between the blood cells, you can see the Staphylococcus also, and this is called as sepsis. So sepsis is a kind of blood poisoning, and if sepsis happens, it's very dangerous, and people usually die if they don't get a proper medication and proper diagnosis. And abscess. This is the collection of pus that builds up in a tissue, and the sign of abscess include redness, pain, warmth, and swelling. So you can see this uh, redness happens. You'll find pain also, and the site will be warm sometimes, and you can find the site will be much more warmer than other parts of the body, and this swelling. But this part is inflated now. Okay, so why it is inflated? Because this site is infected by one pathogenic organism. It is called Staphylococcus aureus. When it infected, our body try to remove this bacteria. So what the body do? It surrounds the bacteria like this and puts this immune cells to inside to kill the bacteria. So that this bacteria will not able to spread outside and go to the other parts. So for this purpose, body make this defensive function and protects our body. But this is the sign and symptom. We call this is the symptom and sign, but this is in really this is the immune defense of our body. So if why it pains? Why any this kind of things give pains? Because pain is also a defensive factor. So when there is pain, that means body say that this place has problem and please don't touch or don't disturb the place and take care. So we get the pain. I previously I told you about this, why pain happens to us in any injury or any infection, because that is a immune function caused by any infection or other immunological problem. So our body try to protect us, not to disturb the site. If you will disturb this site, there will be injury, and this bacteria cell will come out from here and will go to other sites, and it will infect more. That's why body try to surround from every side so that this bacteria will not get any nutrient from the body. They will not get any nutrient, minerals, and food. And after a few days, they will develop the stationary phase and they will develop the lag phase. After the lag phase, they will automatically die. So this is the general mechanism of body, how to protect itself. This Staphylococcus aureus has a small genome and it's contain around 2.8 MB circular chromosome, and the guanine and cytosine content of this S. aureus is very low. It is only 32.7%. And the genome also contain this Staphylococcus aureus genome also contain prophage genome. So usually this bacteriophage always infect the bacteria. Okay. For like this coronavirus infect our this one kind of cell, this bacteriophage infect the bacteria, and this bacteriophage release its genetic material into the bacteria, and this genetic element get inserted into this bacterial genome. So when you sequence, you will find the genome sequence of this virus as well. So it also contain this Staphylococcus aureus genome also contain prophage, uh, genetic material of prophage. And the genes that govern the virulence and antibiotic resistance are usually carried using plasmid and bacteriophage. So here you can see two different kind of genetic material in a bacterial genome. One is a circular chromosome and another is plasmid. There is a circular chromosome, and in other places you will find the plasmid. The plasmid contain antibiotic resistant gene. So whatever the antibiotic resistance developed by the bacteria or anything, in bacteria, they are governed by the plasmid. In the plasmid, these antibiotic resistant genes harvest, and but in this bacterial chromosome, you will find other kind of genes. Those are associated with this their metabolic growth and other functions. And Staphylococcus aureus is a gram-positive bacteria. It's a cocci, and it's a round shaped, and most strain of this bacteria of Staphylococcus has the potential to form microcapsule. That means they form a capsule-like structure around them to protect themselves. And the Staphylococcus produce 11 different kind of microcapsules that differ in their chemical composition. You understand? Even if this same microorganism developing 11 different capsules to protect themselves, their chemical composition and chemical composition of these microcapsules are very different. Okay. Do you have any doubt till now, or it's okay?
Okay. Okay, Doctor.
It's okay.
Okay, that's good. If you get any confusion, please ask me if you don't understand, okay? Because this is a little bit new thing for you people. So even if they contain 11 different kind of capsules, this microcapsule, the microcapsule are not similar. Their chemical composition is not same. Their chemical composition is little bit different. So you see here, how this plasma membrane and structure of Staphylococcus you can find here. So it mentioned which kind of chemicals this Staphylococcus releases at a different phase of its growth, and you can see different structural proteins are present outside the cell, and these are called protein A, elastin binding protein, collagen binding protein, fibronectin binding protein, and clumping factor. Okay, so this is the clumping factor. If we'll enlarge this clumping factor, you can see one is cell wall anchoring domain. This part of the protein is associated with the cell wall anchoring domain. This is the repeats. They are similar things. So it is repeats, and this is ligand binding domain. Sorry. So when any molecule comes in contact with this ligand, this this domain capture the ligand, but it should be its own. I mean, it should be a correct ligand. It cannot bind anything. Well, okay. And if you will see the cytoplasmic membrane, this side, you can see penicillin binding protein. So when someone give antibiotic, how it acts. So they contain penicillin binding protein. You can see in the plasma membrane side. Okay. And this is the peptidoglycan layer, and this is the capsule. So we are talking about capsule. In the capsule, you can find little bit of lipoteichoic acid, cross-linking peptides, but this beta lactamase one enzyme. You see, you specifically remember this beta lactamase. It will be very important for you. Okay, this beta lactamase enzyme is present in the cell wall, and ribitol acid. So these are a typical structure of Staphylococcus aureus bacteria. This is the cell wall, the plasma membrane, and the capsule layer. So we are discussing about the capsule, and you can see this uh, clumping factor. Clumping factor is coming out of the capsule, and you will find a beta lactamase enzyme in the cell wall. So the beta lactamase is a very important enzyme secreted by this bacteria to neutralize these antibiotics. So the role of beta lactamase is very important in the medical biology, sorry, in the in the field of microbiology. You need to understand about the beta lactamase, what it do, and how to deal with the beta lactamase producing bacteria. And almost all bacteria produce beta lactamase, which has potential for antibiotic resistance. So in different growth phases, how you can deal with the different proteins or toxins or enzymes. So this cell surface protein, this is the cell surface protein. You can see protein A, this clumping factor, fibronectin, or elastin binding protein. Okay. Many of these proteins are made during the exponential phase of bacterial growth. So when the bacterial growth is very exponential, Staphylococcus aureus produce this cell surface protein, and many act as adhesins. So do you know what do you mean by adhesins? Anybody know what is the meaning of adhesin?
Of what, doctor?
This one, adhesins.
Yes, we know.
Attract to the other.
Attach.
Okay, good. So this is one protein. Okay, adhesin is a protein, and it helps to bind to any surface. Okay, and also animal cell also produce this kind of proteins. So when uh, people do this cell culture, this in the culture media, this animal cell also produce the same cell surface protein adhesin, and cohesin also. There is another protein cohesin and adhesins. They help to attach the surface. So they will attach to any cells or any tissue or any organ. So once it attached in some places or in any cell, tissue or organ, it will start to multiply. Hmm. And it will lead to its exponential growth. Toxins are usually secreted during the stationary phases of bacterial growth, and this toxin helps to damage the tissue. Whatever toxin bacteria is produced, this toxin usually always help. You see, these are the toxin. This is alpha toxin, this is TSS1, sorry, TSST1, and this is enterotoxin. So what this toxin do? They will help to damage the cell, nearby cell, or in the same cell where they are growing. Okay, so that they will damage the cell, damage the tissue, and will enter inside and continue to replicate. And in third case, Staphylococcus aureus produces enzyme, and these enzymes are proteases, lipases, proteases and lipases to invade the tissue and spread to additional site. So this Staphylococcus produces this cell surface protein. It attaches to other cells so that it will other in some places. It produces toxins and they help to damage the tissue, and also it produces enzymes like protease and lipase to invade the tissue and spread the additional and spread to the additional site or other sites. So what is the meaning of a protease? Who can say?
Proteases.
Yes.
I think enzyme, type of enzyme
Yes.
that break down the protein, maybe.
Yes. Not maybe. Yes.
Yes. Okay.
And what is lipase?
Also a type of enzyme break down the lipid.
Yes. Yes. So lipase, protease, and these are the enzymes which break down the protein. So when this bacteria infect any cell or tissue, in our cell we have this protein and lipid. You know, this our plasma membrane is bilayer, and it contains lipid and protein, right? So it need to break down the protein and the lipid of the plasma membrane so that it will able to enter inside. So this bacteria secrete this protease and lipase enzymes, and this protease and lipase enzyme damage the plasma membrane of the host cell, I mean the infected cell, and they continue to grow like this, and they continue to do like this. So that's why they are very dangerous. These all these cell surface protein of Staphylococcus aureus act as virulence factor. So all these cell surface protein of the Staphylococcus aureus act as virulence factor, and And what these virulence factor do? For example, protein A. You can see here is protein A. So this protein binds to the antibodies. So if it infected any tissue or cell, this protein bind to the antibody of the infected organism and prevent phagocytosis by immune cells. So the antibody binds to this protein, and when any antibody binds to this protein, it don't undergo phagocytosis by immune cell. So when protein A binds with any antibody, this bacteria almost became very strong, because phagocytosis cannot happen. Immune cells cannot degrade it, and it will make the problem. And there are coagulases. There is another enzyme that causes the coagulation of plasma protein. So if this Staphylococcus contain this enzyme, it makes coagulation of plasma protein. So it will damage the shape of the plasma protein. So do you know what we mean by coagulation? Sorry, coagulation. How many of you know about the coagulation?
Could you repeat it, doctor? What is it? What is it?
Coagulation. This means. Do you know what is the meaning of this?
No, doctor.
Okay. I will tell exactly.
It is a disease when the bacteria, when the blood become coagulate.
Coagulate means bringing together and make mess like. It will not be free. It will be a kind of mess like structure. It will make.
It becomes,
This enzyme will stick everything and bring together, and coagulate everything.
yes, we understand it.
Yeah. So it is in the context of that. And when everything will get coagulated, they are not in their original shape and size. So it cannot function. Plasma protein cannot function. So it will make us problem, right? And this clumping factor binds to the platelet. You see how dangerous this platelet is supposed to provide us immunity in our body, right? This clumping factor binds to platelets and help the bacteria to grow at the wounding site. So when the platelet come to this site, it binds the platelet and it reduces our immune system. So how they develop their system very cleverly. They coagulate plasma, this plasma membrane, this protein A binds with antibody and don't allow to get scavenged or get degraded by phagocytosis. This binds the platelet and reduce our immunity and allow the bacteria to grow. They are very clever kind of organism.
Doctor,
Yes.
are these types of protein found in all bacteria surface?
Not in all bacteria. Every bacteria have their own different kind of things. Hmm. Here
Different kind of protein?
yes, different. For example, in a mycobacterium, we have something different thing, mycolic acid. And the secret more mycolic acid, and that damages more like this. So different bacteria have their different function. So they 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 have their mm, enzymatic and other function according to their requirement. So how staphylococcus need and what need and when need to get infect organism and to stay there and to grow there more frequently, it depends upon their genetic content, and we cannot say that every bacteria have a same thing of enzymatic and protein component.
Not same exactly. I mean same types of protein.
Yes, repeat.
Not same function. I mean
Not assumption.
not same function. I mean all type of protein. Not all. Some type of protein has same this type of protein. I mean bacteria. You understand me?
No. You will find some common thing in every bacteria. Okay, that's why they are bacteria. Hmm. Some common thing, for example, you will find this lipoic acid, eh, this uh, the cell wall components, and other things, few uh, RNA, sorry, few polymerase, few transcription factors. There will be a lot of similarity, but when comes to a pathogenic thing, when come to the concept of disease causing and dangerous activity, on that case they are always different. So only one or two protein or enzyme make a difference to cause a different kind of disease. For example, we get this flu every year, flu, but it don't make us any problem. Hmm. And even in 2 months or 3 months, we every time get flu. But the same the coronavirus, the same category. It has different genetic content and it is causing a different problem. It has different protein, but in few cases, up to certain their genetic content is similar, flu-like. So when there will be and that's why you will find different symptom for different diseases. You will not find same symptom.
Okay, doctor.
Okay. And that's why there are different diagnosis procedure. We cannot this now these days we get these small kits. We put little bit of blood and we check whether infection happened or not. And every kit has different antibody combination because they produce different this extracellular protein. So when they produce different extracellular protein, obviously the protein, the antibody will be different. So we need to make different test kit. We cannot use the same test kit. If they would be same, we would use the same test kit for diagnosis in the hospitals. Okay.
Yes, right.
This Staphylococcus aureus is also a pyrogenic bacteria, and this toxin or exotoxin, and you can say super antigen that activates immune cells and cause excessive inflammation. So we see this, we already seen these abscesses. So what they do, This exotoxin is very hyperreactive, and you can say super antigen. And when the super antigen, they release the super antigen, the immune cell of the body get hyperactivated, and therefore it produces excessive inflammation, and we get these abscesses, round swelling things, due to excessive inflammation. So any organ, if you get any such kind of abscesses in any infection, that means there is one super antigen. The pathogen has produced this toxin, or you can say this toxin is super antigen, because it is hyperreactive and making this swelling. And these super antigens are class of antigens that result in excessive activation of immune system. So when they produce this, for example, coagulase or clumping factor or anything, they activate the immune system very high. And more specifically, this immune cell T cells, they produces the T cells, and resulting polyclonal T cell activation and massive cytokine release. So when they produces they are this exotoxin, our immunity get hyperactivated, and it activates non-specific activation of T cells. So there is usually activation of this immune cell. There is one T cell, one B cell, helper cell, macrophage, all are there. You might have known about this lymphocyte. So this is this T cells, and it produces polyclonal antibody when it get activated. And when this polyclonal antibody is produced, our body also releases a lot of cytokine. That is also one immune signaling or immune protein, you can say. And the alpha toxin also released by Staphylococcus aureus, and causes pore formation on the surface of host cell, leading to their death. So you see, Staphylococcus also produce alpha toxin, and what this alpha toxin do? It causes a pore. When there will be alpha toxin, it will causes a pore on the surface of host cell. And if this pore formation will happen in any organ, for example, if Staphylococcus grow in heart, and if there will be pore in the heart, what will be the situation? We will die immediately, is not it? Are you there, students? I cannot hear anybody.
Yes, we are here, doctor.
Okay. And please put this ID number for your attendance in this chat box. And there are only few whoever may have forgot to put their ID. Please put your ID number. So this alpha toxin is very dangerous, as you know, and almost you see what they are doing. It's producing super antigen. It's producing alpha toxin. It's producing also enterotoxin B. So when if you will eat any food having this Staphylococcus aureus infection or contamination, it will also lead to severe diarrhea, nausea, and intestinal cramping. So in your abdomen, you will find a lot of cramp. if you have taken this contaminated food, and if there will be staphylococcus, because they already have produced this enterotoxin B. Hmm. And this enterotoxin B causes food poisoning. And this is a very dangerous kind of uh, toxin. And you know about the toxin, we already studied about exotoxin, endotoxin. And this is enterotoxin. Uh, and enterotoxin, we are calling this enterotoxin. It usually bring problem in our intestinal, this colon region, and this food poisoning brings a lot of cramp. And so, you know, how dangerous is this staphylococcus. So it has all its uh, mechanism, how it can suppress you or bypass you or suppress our immune system. They have made all of their, you see, coagulase will coagulate everything, and this uh, ligand binding domain of this clumping factor, it will bind to our this platelets, so it will reduce all the platelets. And it will not allow the platelets to work, because platelets are very helpful for our immunity. So it has developed its system. So they are much more clever than us. We are doing research in the laboratory how to fight them, but they are not doing any research. They are just changing themselves how to fight their host. I mean, the uh, person or organism where they will grow. They are very clever than us. They are just changing their genome and genes, so that they can acquire their resistant capacity more effectively and uh, more frequently, so that they can attack and continue their growth and development in the host tissue. So I think I should stop here, because this is a little bit of complicated text and a complicated lecture we are going through with a lot of new things. So we'll next continue from slide number 11, and I will stop here. Okay.
Okay, thank you, doctor.
Thank you. And if you anybody have any doubt, please feel free to ask me, and I will be very happy to make you understand as per my potential. Okay.
Okay, doctor. Thank you.
Okay, doctor. Thank you.
Okay.
Thank you, doctor.
Thank you. Thanks.